So thank you, Maha. I think Raja, I think you're up next. And so what we're going to do is hold the questions at the end when we have a panel discussion, much the same way we've done before. Thanks.
So how do I pull up the slides? Oh. Does this work? All right.
You're holding it the other way. Yeah.
Oh, the other way around. Okay. I need my glasses, I guess. Okay, great. All right. Thank you. Thank you, Marco, for inviting me, and it's a great pleasure to be here. And there was a really lively discussion that happened in the other room, and it's really exciting. I do not belong to the TB community, but thank you for Marco. I have a background in microbiology, and I'll talk a little bit about how and what we do, and maybe there are opportunities that we can work on together. So I am the PI, along with Dr. Simonian from FDA, on a platform, a bioinformatics platform. And the bioinformatics platform space actually is great if you are into development of algorithms, development of interfaces, and development of standards. And all of these things actually are very much interlinked if you're trying to do some analysis which you want to take it all the way to the FDA, for example. And I'll show why it is important. But there are many things that still need to be done with the community to take it to that level. So we are pretty much agnostic. We work on a bunch of different things. We work with exome, RNA-seq, DNA. Basically, the platform has tools to handle different types of data, glycomics, proteomics. We build knowledge bases. We are big into ontology and standards. And we also work with the community to come up with representative sets, representative genomes, representative data sets which can be used. And of course, in terms of sharing, we are also or try to be in tune with the community in terms of APIs and other standards and so on. Now, Hive is a platform, and there are a few key features. I mean, we have actually a lot more to say about it, but I just want to kind of go at a very high level. It's a distributed storage and computing system. It has its FISMA moderate equivalent setting, and it has FDA, industry, and patient data in mind. It has been approved to authorize, ATO approved, means authorized to operate in a regulatory environment. So there is an installation of Hive inside of FDA, which handles regulatory submissions, if any of the regulatory scientists want to use it. It has granular sharing capabilities, which is extremely important. And of course, it can handle terabytes of data. It can really handle large volumes of data. And the compute speed also is important. And we work in it. We actually develop a lot of visualization tools, because one of the things is that when you're looking at diverse type of data, you need to be able to visualize it to make decisions. So we really invest a lot in developing these tools. And my group, as is at George Washington University, and the FDA group is at FDA. Together we are on 28 people, and we have joint lab meetings. We have a research collaboration agreement with CBER, FDA, and so on and so forth. So I have some slides of some examples which I thought could be of interest to this community. There is one algorithm where we have a SNP-based clustering and phylogenetic analysis, and a lot of this is published actually. So when we come up with a new algorithm, we make sure we publish it in the open domain so that people can read on it, and if they want to use it, they can use it. Here is an example actually which is used on the Hive platform for HIV analysis. So it's a clonal algorithm, so there's heterogeneity in HIV, HCV, and many of the other viruses, and many of the bacterial genomes, and including even cancer. So trying to use NGS data to tease out this clonal diversity is a challenge. So the tool that we developed and the algorithm that we developed can do it quite well, and we have been doing this with Duke University and a couple of other groups on a regular basis, and the results look very, very promising. So you put in sample reads and the reference strains. Here we just do 12 here, but you can have hundreds of reference strains. You run QA/QC, and end of the day, you get on the top left, there are there are some graphs which shows recombination and clonal diversity. Here's a real example, a stool sample of enteroviruses, and here you see that there is a lot of diversity in this particular sample, and we have many, many examples, real examples like this. We also develop metagenomic analysis. There is interest within regulatory scientists and elsewhere to understand what is in the metagenome. There are many algorithms out there. The limitation is the data set. So most of the algorithms, if not all of them, use a reduced database because computation is expensive. So we have developed an algorithm based on subsampling and iterative subsampling where we can come up with a reduced set based on the subsampling method. Then we can use deeper methods to identify exactly what is in the metagenome. And we have been doing this. We have actually, this is a full project where we actually even collect samples to showcase this works from people, and then we sequence them, and then we analyze them, and we are creating this DB of potentially what self-reported normal people, like they're healthy people, say that this is, I'm giving fecal samples, I'm healthy. We are putting it in a database to get an understanding of what is the diversity within the healthy population. In the past, we have done this for the Army, rapid pathogen detection. Again, I'm not going to go into a step, but basically here we have unique signatures from different pathogens and trying to use that to understand very quickly, what is in a data set, and go to pathway analysis and so on. So these type of detection pipelines requires really high quality databases. And we work very closely with NCBI, EBI, folks over in Australia, Japan, and so on and so forth to create high-quality reference data sets. So I will spend a couple of minutes on this slide and most probably the next slide to talk a little bit of the biocompute object. So all of which I said, these are just algorithms and software. Am I doing some magic behind it and giving you the result? Last week, we had around 200 people in an FDA and GW-sponsored workshop. In that workshop, what we tried to do, although our work started in 2014, there was the first workshop in 2014. It took us to 2017 to get to the stage that we were last week. In this workshop, we got together and we asked the question, can we come up with a good mechanism to communicate our computational pipelines to FDA? What did I do? Which software did I use? What version? What parameter? Are there any validation kits? What was my input and what was my output? Are there any validation kit that the FDA could run or anybody could run for that matter? And we went through a lot of things. So here is what we have come up with. This didn't happen just at the workshop, by the way. We have been working on this. We actually have a paper which describes this concept. So what we are saying here is that if we can capture, in the middle you see alignment, variant calling, and mutation profiler. That's what people talk about when they are putting up slides and saying, okay, this is what I'm doing. But we want to capture the complexity filter, the amino acid calls, what is the entropy, and so on in the parameterized domain pipeline. At the bottom is the input domain, what is the input and what is the output. And then on the right-hand side is actually also, or in the middle bottom is the error domain. So when you have all of these pieces of information together, then you create what we are calling a biocompute object. And all of this information can be written up in a human or a machine-readable format. So what is that? So this is a JSON file. Even though for some of you, this might be a little bit too busy, but a JSON file can easily be read by a lot of parsers and produced like a regular text file. So currently, based on what we heard during last week, when people submit computational pipelines to FDA, they submit a PDF or a journal, right? You write it in Microsoft Word or something, and you submit it. Sometimes it has too little information. Sometimes it has way too much information. So what this is trying to do is that work with the community to really try to say, okay, these are the pieces of information that we want to put into the biocompute object. I want you to focus on that bolded third line, name. So this was done by one of the FDA regulatory scientists, Eric Donaldson, actually. So in this particular case, he is trying to say HCV1A, ledipasvir, resistant SNP. And if you notice, we are actually within square brackets. In the actual JSON, we may not need to use square brackets for those of you who are familiar with JSON, but just for representation. So HCV1A is a specific thing I'm talking about. So we put the tax ID in there. And of course, this is all menu-driven. And then ledipasvir as a PubChem ID. So it's not just free text. There's an ID behind it, which means that if there are changes in the name, or if I want to find similar drugs, by looking into PubChem, we can do a lot more things. And then the resistant SNP is a sequence ontology, and so on and so forth. And then underneath, there's a usability domain, and so on. And for some reason, something is not showing up in the workflow, but that's OK. In the parametric domain, it tells you what are the parameters that were used. So having this common method, and we have tested this, actually. If a person has a good knowledge about the pipeline they are using, it takes half an hour to write this one up. But most of the time, one person does not have knowledge of the pipeline. So when we approach a person and say, can you please tell us what pipeline you're using to do this? They have to go back, and they come back after a month. Oh, now I found out all the details. So that information is not there. So what we did is that we invited Galaxy, CLC Bio, DNA Nexus, 7 Bridges to the workshop last week. And what they will be trying to do, or they will try to do, is that when you run a pipeline in these platforms, and including Hive, and Hive, we are already doing this, it will spit out a biocompute object, which can be read, or it can be put into any software that is compatible, and it can produce the results using the verification kit. So this is the idea that we have proposed. And again, this is very early stage. We are collecting input, and we'll be more than happy. I'll be more than happy to talk to you, or if you want to email us, we would want to listen what you think. So there were many people who came who gave talks and were part of panelists. I mean, this is a snapshot of the folks that were involved. We're also working closely with the standards folks, Fire Genomics, GA4GH, and so on and so forth, CWL. So we are really hopeful that there will be some community consensus behind this. And our overall Hive project, I would want to definitely acknowledge our funding sources, NIH, FDA, GW, NSF, and pharma. And special thanks, some of the things I showed, special thanks to some of the folks up there. And once a year, we do our Hive Jamboree. So this is 2016. So thank you for taking the time to listen, and I'll be happy to take questions during the panel.